for the introduction, and I'll start with something simple. All the humans who are here, and those who are listening to me, and those who are still glued to their computer and mobile phones, and all other life forms are made up of basic constituents, which are building blocks called cells. And the eukaryotic cells, which are ours, are subcompartmentalized into different organelles. And one such organelle in them is lysosomes. And lysosomes have primary function to catabolize the biomolecules and recycle them, especially in conditions like now where you are starving and really cursing me to take time before you go for the lunch. And all this is achieved in the lysosome through a set of hydrolytic enzymes which are present into the lumen of the lysosomes. And the outer membrane, now the limiting membrane or the lipids present on the outermost layer needs to be protected from its own hydrolytic enzyme. And protection is achieved by coating it with a sugar layer called glycocalyx. And the lipids, which are supposed to be degraded in the lysosome, are delivered on these vesicles called intralysosomal luminal vesicles. As I said, in the lumen, all the enzymes are soluble in nature, and they somehow need to bind and selectively recognize ILV membranes and dock and selectively degrade the lipid cargo. And this is achieved by one key component at ILV membrane, which is called BMP, not the protein, but a lipid, which is called bis-monoacylglycerophosphate. And through its various electrostatic and structural interactions, it helps stabilize these lipases so that they can degrade the lipid on the cargo. The two questions we have been interested in is how this is synthesized and how it's stabilized in the lysosomes. BMP was discovered half a century back, and it exists in our systems, in our physiology, and in our body, but we didn't know for all these years that how it is made and how we acquire it in our living systems. And the second question is the stability. I'm talking about hydrolytic lumen, where essentially everything should be degraded because the primary job is catabolism, but somehow this is stabilized, and it's interacting with another lipase. It's a lipid. It's not being degraded, but it's doing the devil's job of helping it degrade other lipids. The stability part was quite easy, and it is offered by unique stereochemistry. If you look at the rough structure here, it's a lipid. It has glycerol, phosphate, glycerol, and fatty acid appended to it. And the glycerols are in ST stereo configuration. This is in contrast to every other phospholipid present into living systems or eukaryotic systems, which are in R stereo configuration at glycerol. Now, pardon me with this jargon, but what I mean by RNS is just the 3D orientation of a molecule depending upon the asymmetric carbon present in here, and good example is our left and right hand. And over the billions of years of our evolutionary history, we decided to choose one hand over the other, like amino acids exist in L form, sugars exist in D form, not the other form. So it makes BMP stable. And my last slide is we were trying to map how it's synthesized, and we used several complementary approaches, and we figured out two enzymes in the lysosome, which are PLD3 and PLD4, which do a transphosphorylation reaction and synthesize this lipid. And we investigated what it means for the physiology if this lipid is missing, and what we find from our preclinical and cellular models, that if this lipid is missing, you end up having lysosomal storage disease or neuroimmune functions. If you want to know more about it, please stop by at poster 11. Thank you.